overusing glyphosate on your crops. Have you not heard about the harmful effects they're bringing?
I've been in this industry for 20 years. You don't say so much and go play your video games.
Excessive use of glyphosate has caused many diseases in crops. This harmful herbicide is found in soil, crop products, animals, freshwater, and even people. Embryo mortality in animals has dramatically increased due to the excessive usage of glyphosate. Moreover, the accumulation of herbicide in surface water may cause water pollution.
Glyphosate, a non-selective and post-emergent herbicide known to control more than 150 weed species. It is a popular herbicide among farmers due to its broad-spectrum weed control. We have multiple professionals with us today to give us more information on the situation. Dr. Ping, would you like to explain how the excessive use of glyphosate has impacted the environment?
Good morning, viewers! Were you wondering how glyphosate causes diseases to develop in crops? Well, I'm here to share a bit of information about it. This herbicide prevents the synthesis of amino acids through the binding and blocking of EPSPS activity. This enzyme is vital for the synthesis of certain phenolic compounds that contribute in plant immunity. Moreover, glyphosate causes plants to stop synthesizing defense compounds, which causes crops to become more vulnerable to soil-borne pathogens. Research has shown that overuse of glyphosate could lead to diseases such as fusarium head blight in agronomic crops.
Thank you, Dr. Ping. Are you also able to elaborate on how glyphosate may be of potential harm to animals and cause water pollution?
Yes, I can actually. Animals are highly likely to be exposed to glyphosate through their feed. For a fact, recently, researchers have discovered multiple harmful effects of dietary GBH exposure in chickens. These effects include embryo mortality, oxidative stress induction, and microbiome alterations. On the topic of water pollution, glyphosate accumulation occurs mostly in the upper parts of soil layers due to its high affinity to soil particles. Hence, the herbicide is easily carried to groundwater, surface water, and water sediments. In 154 water samples obtained from Midwestern US, 36% of them contain glyphosate. It has been reported that this chemical may present long-term and far-reaching risks in natural environments.
Once again, thank you for your input. Dear viewers, a common method to solve the negative impacts of any pesticide through bioremediation. Bioremediation is a biological process that uses living organisms, mainly bacteria, fungi, and plants, to degrade, remove, alter, and immobilize and detoxify waste products and pollutants from soil and water.
The positive view on bioremediation stems from how it takes advantage of the natural ability of microorganisms to extract chemicals from water, soil, sediment using energy from sunlight. Moreover, it is a cost-effective technique compared to physiochemical treatment methods. Lastly, bioremediation requires less energy and often requires little to no residual treatment.
Today, we have invited Dr. Pong to address the current ways to bioremediate this harmful herbicide known as glyphosate.
To sustainably remove glyphosate from the environment, researchers have decided to look into microbial degradation. As for now, we have pinpointed two main glyphosate microbial degradation pathways, namely the AMPA pathway and the cyclosine pathway. I will first describe the AMPA pathway. After the carbon-nitrogen bond cleavage in the glyphosate molecule, it results in the formation of AMPA, which is then metabolized to methylamine phosphate or phosphonoformaldehyde. Ultimately, they result in the formation of formaldehyde. Next, we have the cyclosine pathway. This pathway involves the cleavage of the carbon phosphate bond in the glyphosate molecule, producing a phosphate group and a cyclosine, which is then converted into glycine and formaldehyde.
Thank you, Dr. Pong. In recent research, bacteria and fungi have been reported to be able to effectively degrade this harmful herbicide. Hence, we have invited Dr. Ding and Dr. Dong to give us more insight on that.
Thank you. So, in the AMPA pathway, it is found that Atrobacter atrocyanus ATCC 13752 rapidly degrades glyphosate into AMPA. The AMPA is then used as a substrate for the enzyme CP lyase, and they will be converted into phosphate and methylamine. These byproducts are then absorbed by the bacteria. As for the sarcosine pathway, Pseudomonas sp PG2982 is used to cleave the CP bonds in the glyphosate. It is then metabolized to sarcosine and further degraded into formaldehyde and glycine by sarcosine oxidase. While other bacteria such as Chromomonas odontothermitis P2 and Lysine-Bacillus sphaericus can degrade glyphosate through cleaving the CP bonds too, but with higher degradation percentage. And that's all from me.
Thank you, Dr. Ding, for the explanation on the bacterial degradation of glyphosate. I'll now pass the floor on to Dr. Dong.
The utilization of fungi is also a recommended way to degrade glyphosate. This is because fungal bioremediation is an environmentally friendly, cost-effective, and feasible solution to this contamination problem. In the AMPA pathway, Aspergillus oryzae A, FO2 is utilized. Just as stated before, the CM bond in the glyphosate is broken to form AMPA and then further metabolized to various metabolites. Contrastingly, Pyrrolinasilum is utilized in the sarcosine pathway. Through this fungus, sarcosine and a phosphate group are released via a CP bond cleavage route. Sarcosine may then undergo further degradation to produce formaldehyde and glycine.
There we have it, folks. I hope this was an informational session for you as it was for me. Thank you!